and then we'll get a pretty good start on 12 as well. Okay, look about right. So we're talking about variation, right? We already talked about one big engine of variation. That was recombination. Now we're going to be talking about the second big engine of variation. That's going to be random assortment. So here's basically what random assortment is. So when you look at basically aligning your homologs on the metaphase plate, what's the probability of, let's say this is mom is red, dad is blue, right? What's the probability of mom ending up on this side as opposed to this side? Well, yeah, it's a coin toss, right? So basically you just flipped a coin. You had equal chance of mom ending up on the other side as on this side. That's true for all chromosomes. So if you basically think about it, they're just showing 3 chromosomes here, right? We have 23. So basically, if you think about how many different combinations you can come up with 23 chromosomes, you're talking about every single event, all 23 events, having a probability of 1/2, right? So that's basically 1/2 to the 23. Okay, so essentially, what you're talking about is 1 over a really, really big number. So the probability of any one event happening or any one pattern happening over 23 independent events is really, really tiny, right? And so what we're talking about with independent assortment is you can have different mixtures of this, right? So for instance, in this particular one, you had mom, mom, mom. In this one, you had mom, mom, dad. In this case, it was dad, dad, mom. And this is mom, dad, mom, mom, dad, 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 mom, dad, and so forth, right? This is only 3. So basically you do this 23 times. And how likely do you think it is that you're going to get the exact same pattern twice? Yeah, really unlikely, right? Now think about this for a second. I think one of my other students, I forget which section it was in, looked it up, and actually it turned out to be like 8.3 million, like 1 in 8.3 million, which is not bad. Okay, it's bad, right? So you don't want to be that one. That's not the one you want to actually rely on, right? So basically, this is one engine of variation. Now, if you think about it, 1 in every 8.3 million basically means that if we got 8 billion people on the planet, there should be a couple of more of you out there, shouldn't there? It should be possible in 8 billion people to pull 1 in 8.3 million a couple of times. But remember, this isn't just, we're not just talking about this individual engine, right? This is already on top of recombination. So remember how recombination went. So when you think about this idea, you're the only person on the planet. You're the only you here. You're the only you that ever has been. And you're the only you that ever will be. Does that sound far-fetched? Initially, yes, right? Considering how many people are on the planet. But then when you start to actually take a look at just how much variation there is and how much capacity there is for variation, just with recombination alone, right? You're talking about like random number generators with infinite capacity to reshuffle everything in your genome. Then you add on top of that, this random assortment, where you're talking about a 1 in 8.3 million chance of getting the same pattern. That's on top of recombination. So if you're asking for the same U twice, what you're asking for is the exact same pattern of recombination that we saw in the first engine, which is nearly impossible. A lot more than 1 per 8 million. It's like, it's an astronomical number, right? I mean, it's an astronomically tiny number. But you want that pattern to happen twice and random assortment to happen twice. So you have an astronomically tiny number in recombination and a pretty darn tiny number in random assortment. You're asking for both of them to happen exactly the same twice. And basically any mathematician worth their salt is going to be pretty much call that zero. Okay. Yes. Is it possible? Of course. But at some point we start to get silly when we start to argue that, you know, it's kind of like Dumb and Dumber, right? It's like, so I got a chance. No, you don't, right? Not in any real world. You don't really have a chance. This number is zero. Okay. And so that's just, we've only gotten two. Remember I said there's three major engines of variation. We've only covered two. Okay, so let's take a look at the events of anaphase. So now that we got everything lined up in double file, along the metaphase plate, a couple things have to happen in anaphase, right? First of all, you got to resolve those chiasma, right? So you got to untangle those homologs from each other. And then when you do that, basically you're going to separate out your homolog, or only the homologs are separated in anaphase 1. The sisters stay together, so there's no resolution of cohesin. Right, that's that glue-like protein that holds the sisters together. There's no resolution there. However, you do have to resolve the chiasma, right? So the chiasma have to break, and you also have to resolve the synaptonemal complex, right? Those two things are what's holding the homologs together. Now, once you resolve those, then you're able to separate out the homologs. And then at this particular point, you will have become a haploid. Remember, in anaphase 1, you're going from a diploid set to a haploid set because you're separating the moms from the dads. So you're reducing yourself by half. Okay, that's kind of what it looks like. So here you can kind of see you've got the same, you know, microtubule setup. With your polar microtubules attached to each other, right? That's your push microtubules. And then of course your kinetochore microtubules, which are pulling toward the poles themselves. So in this case, the only difference is you're pulling homologs. Now, the important thing is, remember that the trick to get this to happen, like the way you get this to happen is different than mitosis, is the way you line it up. All right. In mitosis, it's single file. In meiosis 1, it's double file. Right. So you're basically lining them up side by side. And that's kind of what the secret is in terms of getting this to sort of separate homologs, as opposed to starting to separate out the sisters. So once you actually separate these out, then you just move right into your telophase 1. So you have the same events of telophase, right? Your nuclear envelope is going to reform. Your sister chromatids are no longer identical, right? Why? Because they've recombined, right? So you have either parentals or recombined. And you kind of start to reverse some of the issues of prophase. However, here's the thing. Because this is a two-step system, you may or may not actually go into cytokinesis, right? So it depends on the cell. So for instance, in sperm, you don't need cytokinesis. Because all you do is you just basically just finish. All you do is you just finish meiosis 2. Okay. In eggs, they are arrested. So they need to go through cytokinesis. So it depends on what you're doing, right? I mean, for instance, if you're going to just basically power right on through, just 1, 2, boom, boom, and finish it all out, then you forget about cytokinesis, you just go straight into prophase 2. Okay. However, if you plan on sort of spending a little bit of time hanging out, then you go through cytokinesis so you can kind of like sort of tidy yourself up. And this is really kind of a difference between male meiosis and female meiosis. Sperm are basically made in a batch-wise process. Basically, what males do when they go through meiosis, spermatogenesis, basically is what it's referred to, is they basically go through meiosis 1, and they don't stop at all. They basically just power right on through from meiosis 1 right into meiosis 2, and then they finish and they stop at the end of meiosis 2 when they have their 4 genetically non-identical products. That's when they stop. So for males, they don't go through a cytokinesis after meiosis 1 because they just basically power right on through. In females, however, what happens is they start meiosis, and then somewhere in meiosis 1, they go through an arrest. So they kind of sit there in stasis for a little while, and for a long while, actually. And so at birth, a female has all of her eggs that she'll ever have, and they're all arrested in meiosis 1. Now, those eggs she'll hang on to and she'll have for decades until she hits menopause and sort of she rewires her physiology. But those eggs could sit there for a while. And so as a result, they're arrested. So they kind of like stop and pause. It's kind of like stopping at a rest area when you're on a long road trip, right? So you're just going to hang out there for a little while, and you're going to wait until it's your turn and your turn to go. And that's basically what happens. And so every month, one of these eggs gets called up to go through follicular development. And when they do, they begin to also finish out the process of meiosis, but that only happens once a month. So all the other eggs that didn't get called up for that process are still in stasis. So they're still in an arrest. So the egg that gets called up when you're 40 has been there for 4 decades, suspended and arrested for 4 decades. And so because of that arrest, then what happens is the eggs will go through a type of cytokinesis just to kind of make sure everything is stable and sort of finish everything up a little bit and kind of tidy it up a little bit so it's kind of in a nice state where it can be arrested and paused. Okay, so that's an example of a situation where you might go through cytokinesis after meiosis 1. But for the most part, after meiosis 1, it's on to meiosis 2. So here's your telophase 1. You can see you're basically kind of doing the same things. You're reforming your nuclear envelope. At this point, you still have some issues, don't you? You're haploid, yes, but the problem is you still have 2 of everything, right? You still have 2 chromosome 1s. Can't have that, right? So you're not quite ready just yet. So you need to actually resolve that situation. So you're not quite done. Now, after you go through meiosis 1, then you go into meiosis 2. And here's the thing. Remember, meiosis 2, this is the equational division. And the goal of equational division is to separate the sisters, which is exactly equal and identical to what you do in mitosis. So basically, meiosis 2 is spot on identical to mitosis. So if you're comfortable with mitosis, guess what? You've already got half of meiosis down. As a matter of fact, it's so identical to mitosis that typically I don't even bother really covering it because we've already covered it at length in chapter 10. It literally is the exact same. You line them up in single file, right? So here's your prophase. So you're doing the same thing you do in prophase, nuclear envelope breakdown, chromosome condensation, if you have to, if you haven't already decondensed or not. Spindle migration. And then you get into metaphase, but now notice what's going to happen is, unlike metaphase 1, you are now aligned in single file. And so once you go through anaphase, then you're going to be pulling the sisters apart, where that's what you do for a living. And then once you pull your sisters apart, then you're going to get in telophase your 4 products with the correct number of chromosomes, with the haploid number of chromosomes, all genetically non-identical, which is what your goal was, right? And then, of course, at this point now, you go through a terminal cytokinesis. That way, you're going to be able to create 4 individual products, right? And that's what you want. Now, if you're a male, then you would develop these nuclei into a sperm, each one of them. If you're a female, you develop those into an egg. And that's basically meiosis in a nutshell, right? So you basically have 4 different cells that are haploid, that are genetically non-identical. And in us, that satisfies what we need, right? So basically what we have is we have gametes that are haploid that we can then use in fertilization to be able to reconstitute the next generation. As we learned in the alternation of generations, some organisms will produce their gametes mitotically. We saw that, right? So when you have that mitotic body, the haploid body versus the diploid body, we saw that with the mosses, for instance, right? You have the gametophyte and the sporophyte. Some of those will do that. And when you do that, you're actually able to generate lots and lots of gametes. All right, and that's kind of the idea. You want to produce lots and lots of these because after all, you don't really know like which one of these is going to work. So it's a numbers game a little bit here, right? So if you don't know or have confidence that you have 100% reliability that this is going to work the first time out, then what do? You hedge your bets, right? If you think it's only going to work maybe 1% of the time, guess what? You do it many, many times so that you can make sure it works at least once. Okay. And that's what you see a lot of organisms doing. Now, what happens if you screw it all up? Well, we talked about this a little bit. When you have an error in meiosis, we talked about this in the last chapter, or we talked about this in terms of balance, right? That when you're taking a look at aligning things at the metaphase plate, that you want things to be well attached, right? And you have a balance of tensions. Typically speaking, meiosis needs to be symmetrical. What does that mean? That means you have to have as many chromosomes on one side as you do on the other. Because what happens if you don't? Well, if you don't, then what that means is you're going to get too many going to one side and not enough going to the other. And when you have that, that's what we refer to as non-disjunction. This is a failure to properly segregate in a symmetrically balanced way. What does this produce? This produces aneuploidy. A basically means the absence of chromosomes, right? And so this will produce gametes that either have extra or missing chromosomes. Now, remember what we said early in the 10th chapter? We said that animals do not tolerate missing or extra chromosomes, right? That was the whole point of you doing that whole 4th grade chromosome squash cutout exercise, right? The whole point of that was to basically drive home the idea that these are not well tolerated, that even the most tolerable one, which would be Down syndrome, extra 21, a person there only has a life expectancy that goes into their 50s. So their life is drastically shortened. So in general, it is not well tolerated in animals to have missing or extra chromosomes. So then, if you have a meiotic event that's unbalanced and you create a lot of aneuploidy, which is missing or extra chromosomes in your gametes, guess what? You're going to have kids that are not going to have a tolerable number of chromosomes. Matter of fact, you probably won't have kids at all, right? So this is what will cause that miscarriage in the first trimester. This is what causes, by the way, when we're looking at that chromosome squash, the extra 18, right? Some of you guys had Patel syndrome, or the extra 13. Some of you had Edwards, right? Or the extra 21, or the extra X, right? Where does that extra come from? It comes from unbalanced meiosis, errors in meiosis, where you get non-symmetrical segregation in one of the parents, and you get basically this unbalanced segregation, which causes either extra or missing chromosomes. So this is basically by far what causes miscarriages, most miscarriages in humans. And most of those happen in the first trimester. So let's take a look at what this might look like. Or how can we create these extra and missing, right? So let's take a look at this. So what I want to do is that we have two different states, right? So we have an M1 and an M2 situation. So what happens if we non-disjoin, right, in M1, but M2 is fine, right? So here's what's going to happen. So here's my two chromosomes. Normally I'm supposed to do this, right? Separate those homologs. But in an M1 non-disjunction, what's going to happen? So I'm going to get unbalanced segregation. So they all go one direction and not the other. Now then, if we follow this up with M2, then that'll create two cells with nothing in it. And this one, so they'll line up like this, right, in single file in M2. And what'll happen is you'll get separation here. So you'll get something that looks like this. And then this one will look like this. Right, because you're separating. The problem is they're the same. So what happens is now you've got no chromosomes. This is what's called nullosomy. Nullosome. Null means zero. If you have two chromosomes, it's called disomy. But here's what happens. Let's go ahead and mate these. Let's say this is some sort of an egg or something like that. Let's go ahead and mate these to a normal individual who has their normal haploid set. In this case, what's going to happen is you're going to have a zygote with one chromosome instead of two, right? This is what's called a monosome. This is monosomy. In this case over here, you're going to have a situation where you have three. So this is a trisomy. That's how Down syndrome happens. What about the monosomy? Why don't you ever see that one? Because it is catastrophically lethal. So that one never even gets out of the gate. Okay, so you see this one is slightly tolerable, but this one is absolutely intolerable. Okay, so that's the reason why you never see it. It basically gets subtracted out because of its lethality. Well, what happens if it's a normal M1, but we get a non-disjunction at M2? Let's do that one. Okay. So in this case, what's going to happen is we're going to take our normal M1. And then these will get normally separated into two sides, right? Now what's going to happen is we're going to have an M2 non-disjunction event. So in this case, probably what will happen is one of these guys will be normal. So you separate out these two sisters. So one of these typically will be normal. But this one here will be abnormal. So all of it will go this side and none of it will go on this side. So what that means is then you'll get the two in this one and nothing in the second one. Now, what happens if you mate this to a normal mate? Well, this one here will be normal. This one here will be normal. This one here will be monosomy. And this one here will be trisomy. So you can see half of your kids are going to come out normal. So you notice it's better if you get a non-disjunction event later on, because at least you're going to be able to recover normal offspring. Okay. So you can see two slightly different patterns. but both of them are caused by the same problem, unbalanced non-disjunction, leading to either excess chromosomes or too few chromosomes, and that's a lethal event in humans. By the way, if this is kind of a fascinating thing, like how we get, multiple chromosomes and how we're moving chromosomes around, how we get to aneuploidy and all these different sort of chromosomal numbers, then think about taking the genetics class in the fall, because we actually have an entire chapter where we talk about just chromosomal anomalies and how they happen and how that affects human health and things of that nature. So it's actually a fascinating story, because if nobody takes genetics, then that means I'm going to teach AMP2, which is not a bad thing, but I'd rather teach genetics because I'll just taught AMP2 over the summer. So, okay, so here's meiosis versus mitosis. So side by side. So you can see basically you got similar events in prophase. The only difference is you've got homologous pairing here, right? You've got synapsis and you got recombination, right? That's what really stands apart here. Then in metaphase, notice you are lined up in double file. This one is in single file. Then as you separate, you're separating the homologs. And in mitosis, you're separating the sisters. And in mitosis, you're done, right? In telophase, basically, you're done. And then ultimately, in meiosis 1, you're not done, are you? You're only halfway there. So you've only separated your homologs. Now you have to do the other half, which, by the way, looks exactly like mitosis. So you can see here the exact same thing. Now you're basically lining it up in single file, separating out the sisters, and then now you've got 4 different products. Why? Because you had 2 different products from the first meiosis. Okay. Really, you can think of it. I mean, there's only really a couple of major innovations, maybe 4 at most innovations in meiosis that are different from mitosis, right? I mean, the first 3 are in prophase, right? That's homologous pairing, synapsis, and recombination. Those are the big ones, right? The 4th one is the way they align in metaphase. They're aligned in double file, side by side. That's because of homologous pairing. So that's really the majority of the differences actually that you see in meiosis between meiosis and mitosis. So again, like I said, if you want to understand chapter 11 really well, understand mitosis really well. And chapter 11 becomes quite easy. So a couple of things, first of all, before we actually get to this idea, one thing that I mentioned, because I promised you, right, 3 major engines of variation. I've only talked about 2 so far, haven't I? Well, here they are, right? The first one, well, on this list anyway, independent assortment, right? That's how you line up mom, dad versus dad on the metaphase plate. Number 2 is recombination, how you shuffle your DNA as you're making your gametes. What's the third one? Random fertilization. So the 2 gametes that find each other. Do so randomly. Right. So one thing we've been kind of highlighting is where all the moments of randomization are in this process. So remember that males, when they go through gametogenesis, spermatogenesis, they make all 4 products, and all 4 products become sperm, right? But remember, all 4 products are coming from one recombination event, which is one engine. And then as they go through meiosis, they're going through the second engine, random assortment. And then once they develop into sperm, they're added to a pool of alternate different recombination patterns and meiosis processes with different random assortments. And they're added to the pool, right? So every 4 sperm are a different recombination event and a different random assortment event. And then you add all these together by the hundreds of millions, right? Perhaps billions. And then what you do is you release it into the female reproductive tract. And then guess what? The lucky one wins, right? At that point, what happens is the sperm motility takes over, right? It swims up through the female reproductive tract, through the fallopian tube where the egg is awaiting. That's just been ovulated. And only one sperm will actually fuse and unite with the egg. Once it does, the egg becomes insensitive to all the rest of the sperm. So once the winner crosses the line, then everybody's off. So how do you know who the winner's going to be? Nobody knows. Out of all those hundreds of millions of sperm, only one is going to be the lucky winner. No idea who that one's going to be. It's completely random. Right? So here, now let's think about this. Here's what we need in order to make you twice. We need the exact same recombinational pattern twice. We need to follow that up with the exact same random assortment pattern twice. Then what we need is we need the exact same random like foot race with the sperm swimming toward the egg, and we need the exact same sperm to win with the exact same pattern to win twice. Oh, it's worse than that, because that's just talking about the dad. Guess what? What's the likelihood of that happening? What we just discussed there was what is the likelihood that dad will create the exact same sperm twice in his life? You would need all three of those to come together. You would need exactly the same recombination pattern with the same random assortment pattern. And that winner of those four products, that winner would have to be the exact same winner twice. What are the chances of that happening? Not likely, right? So what does it take then to make another you? Take that probability, which we already know is pretty stretching it, right? And then add that to mom. Because mom has to do the exact same thing. So when she goes through her meiosis, she has to go through the exact same recombination pattern twice. She has to go through the exact same random assortment pattern twice. And not only that, but she has to pick, of all those four meiotic products, typically what happens, this is the difference between females and males. Females don't use all four products. She doesn't make all four eggs from all four of those. What she does is she picks one of those four. The other three, she discards. That one is the keeper. And she puts all of her energy and resources into making sure that is one darn good egg. But what is the nature of that choice? It's random. So her choosing of a particular egg to move forward is random. Oh yeah, by the way, let's keep going with this randomness, shall we? So not only did you pick the 1 and 4 randomly, but now what happens is when it becomes monthly, time for that one particular egg to develop follicularly and get ready to be ovulated so it can be fertilized. Guess what? That choice is also random. So you've got this big pot of eggs when you're born that are in meiotic arrest. They all represent a 1 and 4 choice that was random that you made early before you were born. Now, of that big pot of eggs, how do you know which one you pick every month to move forward for development and ovulation? It's random. So notice you randomly picked from your 4 products, and you're randomly picking from those to move forward to follicular development. So what you would require in order to make another you is not only does dad make a carbon copy of sperm, which we already have stressed out the limits of probability on that one, right? But mom would have to essentially do the exact same thing. Same recombinational pattern, which is nearly impossible. Same random assortment at the same time, which is nearly impossible. Picking the exact one of the 4, which is nearly impossible. And then of that big pot, picking exactly that one of the big pot to go with dad's carbon copy sperm. You make the wrong choice, you're not you again. You've got to pick exactly that one to basically make that choice. So basically, to make another you, it's kind of like asking yourself to win the lottery every day for your entire life. How likely is that going to happen? Are you comfortable with saying never? Mathematically, is it possible? Of course, it's one in something, some huge number, right? But mathematically, it's possible. But would anybody in their right mind say, I've got a chance? No, right? Because that number is so astronomically close to zero that anybody worth their salt would pretty much say, yeah, you know what? You're never going to win the lottery every day in your life. That's exactly what you're talking about. When we say, what would it take for you to happen again? It would literally take winning the genetic lottery every single day. So now my question is there ever going to be another you? No, not even close. So let's kind of wrap this up, right? So basically, when you take a look at meiosis, the things that really make it stand apart are these guys right here, right? So homologous pairing, that's a big one. Synapsis, once you do the homologous pairing, and of course crossing over, which is your recombination. So that's a big one, right? And that is by far the biggest. But since this is a two-step process, there is no replication in the middle, right? This is a one cell division process with two steps to it. So you don't do replication in the middle again. So that's the end of my meiosis. Now, what do you say we put meiosis to work for us and start to unpack it? By the way, just to let you know, I mean, if you're kind of worried, it's like, oh my gosh, I'm going to fail chapter 12 because I don't really understand meiosis very well. Meiosis is the mechanism of genetics, but the way we do genetics in general bio is a little softer for you guys. Like we don't really get into the hairy business of meiosis necessarily too badly in this chapter. In genetics, we get into it a little bit more because we have to understand meiosis to understand more genetic principles. So if you want to know where the genetics class starts because you're thinking about it, which you may be or you may not be, where genetics starts is it starts with chapter 10. It starts with a review of meiosis and mitosis and meiosis, and then jumps right into the content of chapter 12 and kind of expands on it and deepens it. So if you're interested and fascinated in some of these ideas and topics in genetics, and you want more, then you're probably genetics is the class that would give you that more. Okay. So let's go ahead and talk first of all about genetics. So basically now what we've got is essentially this idea of heredity. And so what I did was I just basically kind of built in the mechanism, right? The mechanistic foundation of heredity. And that's kind of what we learned about over 100 years or so. But that's not at all where we started. So where we started in the 19th century was with a bunch of ideas, right? So we have a bunch of thoughts on traits and how they're inherited and how they're moving forward. And so what I want to do is I want to do one of my inserts here. So it helps to kind of frame where we are, right? Because in the 19th century, remember I said before that the 19th century was kind of this nucleation century where a lot of the seeds of scientific thought that bloomed in the 20th century were sown. And so, of course, nobody ever thinks about sowing seeds, right? It's just a big dirt patch, right? So that's not the really cool stuff. It's like we don't really get interested until we start seeing flowers blooming. But this is one of those seeds that were sown. And so one of the things that we want to understand is a little bit is about what we understood. Like what did we think back when this was all coming into play? Because genetics didn't actually come along until this guy right here, our favorite Austrian monk, Gregor Mendel, came in and published his paper and did his work on pea plants. So so what did we know before that? Well, think about it, right? We're talking about thoughts on inheritance. So here's the thing. I mean, did we not know anything about inheritance? No, that's we actually knew quite a bit, didn't we? Think about it. For generations, millennia, we knew a lot about inheritance. How do you know that? Because we have a lot of animal husbandry in human history, don't we? A lot of domestication of animals and crops. So we knew a lot about how to breed animals, and that's basically genetics, essentially, right? How we knew how to breed animals to get favorable things that we wanted. As a matter of fact, one of the earliest stories, and this is fascinating because when you're in a genetics lab, like at UC Davis, which is where I was as an undergrad, and you're kind of like doing all this genetics, you're drawing out different sort of crosses and mating schemes, and you're setting up flies, and you're following their genetic traits and things like that. What's fascinating is actually the earliest description of essentially a relatively sophisticated for its time genetic experiment goes all the way back in description in antiquity. You can actually see in a case of this in literature. At least I don't see anything in like Epic of Gilgamesh or the Assyrian text, but there's a story of one of the old Jewish patriarchs, in this particular case, Jacob. And there's a story essentially that talks about Jacob when he was a young man. And he was looking to get married, and there was a particular lady that caught his fancy. And he had to work for her, right? That was the idea. And he was a shepherd. That's what he did by trade. And so as it turned out, that this young lady that he was attracted to was the daughter of his uncle. It was weirder back those days. Apparently inbreeding wasn't a problem back then. But right, so his uncle said, fine, you can have my daughter. It's like the old school thing. You have to ask permission, that sort of thing. So it's old traditional thing. It's like, yeah, you can go ahead and marry my daughter, but you're going to have to work for me in exchange for that. So after 7 years worth of work, you can have her. All right. And so what did he do? He went to work for his uncle, who has Laban or something like that was his name. Right. And so it's kind of funny because the description in there actually describes something very clever and very sophisticated, because as part of his wages, what Laban was going to give Jacob was all the bad sheep. Right. Now let's understand exactly what we mean by bad. Right. So back in those days, you raised sheep a lot of times for a lot of things. Right. There was just a lot of things that you could get from them. One of them was textiles, their wool. Right. So you could actually use their wool, and more desirable than anything as a textile back in those days was white sheep, because you could dye the white wool very well, and you can make it all sorts of different colors. And so white sheep were by far the more valued type of sheep. Well, here's the problem. Not all sheep were white. Some of them were brown. Some of them were kind of streaked and speckled and spotted and that sort of thing. Those would be considered undesirable sheep, right? So I can't do anything with that because I'm going to get all this polluted, right, with brown spots in it, polluted wool. So these are basically worthless sheep. So here's the thing. Talk about a labor dispute, need for a union, collective bargaining. Laban's wages to Jacob was, listen, you can have all these worthless sheep that are spotted. There's that. How about that? So I'm not even going to give you value for your efforts. I'm going to basically give you a bunch of worthless sheep. And that's the way they saw it. But here's the thing. Jacob was a smart guy. And actually, in all the stories of Jacob, it's very clear that Jacob was a shrewd guy. What he did basically was he said, fine, I'll take your spotted sheep, you know, they're worthless as they are. But what's described after that is a really fascinating description of what Jacob did with those sheep. And basically what he did with those sheep is he basically mated them together. And he noticed that every now and then, not all the time, but at a very low percentage, out would come out a nice little white sheep. And then when he got the little white lamb, he would separate the white sheep from the main stock. And then over time, he would kind of continue to collect these white sheep. And in there, there's this really sophisticated like description of how he did this, right? So he was trying to get this particular sheep to mate with this particular sheep over here. And the way he did that was there was some sort of a stick or something like that. I guess it's like, you know, lamb sheep Viagra or something like that. I mean, something to kind of get them all frisky, but it was like an aphrodisiac. And he would use this to sort of get the sheep to kind of make the way. It was really fascinating. You know why? Because that description is almost identical to what modern day husbandry does. Very little change. And not only that, but it's exactly the same thing that you do in a modern day genetics class. You're mating together two different looks of sheep, and you're looking for specific desirable outcomes, and you separate them out and you move them forward to create a different population. And that's exactly what he did. So he basically did a fairly impressive genetics experiment, which shows a lot of understanding and knowledge of genetics. Even though he didn't necessarily know how it all happened, he certainly knew how to manipulate it. And as a matter of fact, as the story goes, he apparently was so good at it that his uncle Laban actually accused him of stealing a sheep. To which, of course, Jacob's response was, "Listen, I can't help it that you're too stupid to know what you have. So back off or I'm taking you down." Now, that part was paraphrased. I don't think that's actually in the story. That's my paraphrase, but that's kind of how it goes, right? So, and it was a fascinating thing because it literally is a kind of a fairly modernish description, what he was doing, of how you approach animal husbandry and domestication and propagating traits across generations. The reason why I like to tell that story is to drive home the point of what we knew and how early we knew it. Because it raises the next obvious question. If we knew so much about it back in those days and we only learned more as we went on, then what was so special about this guy? Right? If we already knew a lot about it, then what was it? How was it different than what was this? Right? How was this different than anything else? Because it seems like we already know everything. Well, we don't. Let's take a look and see what was swirling around at about this time in the 19th century. Remember, Mendel published his paper in 1865-66. Darwin published his paper in 1859. It bears repeating that when Darwin was doing his work, and remember, Schleiden and Schwann were doing their work in the 1830s, 1839, the cell theory part, right? So it bears repeating that a lot of these scientific ideas that were coming in and sort of starting to drop in were pre-genetics. So genetics in a lot of ways was kind of like after some of these ideas. So you can imagine then the weakness, if you will, of Darwin's ideas. You're talking about a genetic thing without any knowledge of genetics. It's kind of impossible to do that, right? It's like trying to like understand the physics of the universe without having any knowledge of math. Well, you're not going to get far there, are you? Right? And unfortunately, I think that is what that's the reason why the entire evolution discussion went off the way it did for the next 100 years. The reason why it was largely just a big spitball match between two philosophical camps and not really sufficient. It was scientific, but not sufficiently appropriate, right? Because you're talking about a bunch of, like what I call the rock zoo, which is just paleontology. It's like a bunch of dusty rocks. It's like, that's not going to tell you anything because evolution is a genetic story. And you have to know genetics in order to get that. So literally speaking, we weren't ready to talk about evolution the way it ought to be talked about until probably the end of the 20th century. So we're just now there. Unfortunately, we got to sort of undo a lot of the nonsense that we put in place in those 100 years in order to kind of get a really good, clear, accurate view. Let's talk about some nonsense here, right? So some of the ideas that were coming in. There's one that was a holdover, Actually, it's probably pretty much gone by now. What that I call sperm bank. That's the correct name. This homunculus is the actual technical name for it. But this is an idea that suggested that in the head of a sperm is this like little itty bitty kid, baby, probably a male. That's just really tiny. And all you have to do is just plant that, and then it'll grow and it'll get bigger and bigger and bigger. So there was a version of this also for the egg. And there's also a version of this for just about everything. Right, this is kind of the way we saw it. We thought there was like a little itty bitty tree, a fully formed tree and a seed. And then what happened was that basically just sort of took root and it sort of became bigger and bigger and bigger. Now, obviously, this is crazy talk, right? Probably born out of bad technology and just a lot of hallucination. Because you've probably been under the microscope long enough to know that if you stand there too long, you're going to start talking to yourself, right? And arguing with yourself. So basically, this eventually got cleaned up because we were able to see sperm actually a little more closely, and our better technology kind of eliminated this one as a possibility. I always like to put this one in there because we have limited technology as well. I mean, it's great, but it's limited, right? Eventually, just because of technological advancement, generation after generation, we will get better technology. We will be able to basically take a look at our current ideas and think, wow, you guys are stupid. Right? It happens every generation. This is just an example. As the technology gets better, we'll then look back as that society and think, how did you guys ever believe in this? You know, but that's because we didn't have the technology to get there. The other one is spontaneous generation. Remember that one? This is basically the living things arise from non-living matter. Does that sound crazy? A little bit, right? One of my high school biology teacher once kind of flippantly said, yeah, this is like if a rock looks like a frog, it's going to turn into a frog. High school and all of its deficiencies, its many, many deficiencies. No, it's not that easy. You know why? This sounds preposterous, doesn't it? Have you ever seen something that's non-living turn into something that's living? Ever in your life? Ever in your life? No, it's preposterous, right? It violates our entire concept of reality. How then could somebody believe this? By the way, not only did some people believe this, but this one persisted actually well into the 19th century. This idea actually didn't get debunked until Louis Pasteur finally debunked it, the father of microbiology. And Louis Pasteur, of pasteurization fame, what we do to our milk, right? Louis Pasteur had to invent sterile technique in order to debunk this one. So this one persisted deep into the 19th century. And I can guarantee you that at the time Mendel came around, there were probably still some very educated people who were still in the spontaneous generation camp. Because whenever you debunk an idea, it takes time for it to work its way out of the system. Okay. So why then would so many educated people sign on for spontaneous generation? It's so obviously dumb, right? Oh, is it? Remember, our ideas are a product of what we know, but more a product of what we don't know, right? It's fair ignorance. Think about it for a second. Let me take you on a hike. It's a great day today. It's a great day to go on a hike. So we're going to go on a hike, and I'm going to stop by this giant boulder. And on it, I kind of see some greenish colored, kind of crusty looking stuff on the rock. What am I looking at? Not moss. It's a little drier looking. Because moss would kind of be a little bit more fluffy kind of wettish looking. This is like literally like crusty. I know. Let us off the hook. You're looking at lichen, right? So basically lichen is a symbiotic relationship between two living organisms, one of which is algae, so it's photosynthetic, right? But it's a living system. There's three different forms of lichen. There's crustose, which is that crusty one that you see on rocks, right? That kind of crusty green on rocks. So that's crustose. And there's foliose, which kind of looks like it has little leaf-like structure. And there's fruticose, which looks like it has little fruiting bodies on it. Okay. But here's this crusty living system on this boulder. Now, if you take a look at lichen on the boulder and you look around, you don't see it anywhere else, especially with crustose, right? So basically what's going to happen is you look around, and let's say you identify it as something living. First of all, that's a bit of a stretch, but say you identify as something living, and you start to look around. And you're like, okay, wait a minute. I'm looking like on the side of the rock. I'm looking on the ground. I don't see anything. I'm looking kind of around, maybe on the trees. I don't really see anything quite like this. So where did it come from? I see no evidence of it anywhere else except for right here on this rock. So if you think about it, well, maybe somehow this rock transformed somehow into this lichen system. Now, are you laughing still? Seriously, come on. A rock transforming into lichen? Are you kidding me? Well, wait a minute. Wait. We're all intellectuals here, right? Let me cite you an example. I've seen similar types of transformations before that seem comical. Think about it. I've seen a caterpillar squirming around, looking nothing like ultimately what it becomes once it goes into its cocoon and gets broken down, and then out comes what? Yeah, does the butterfly look anything remotely like the caterpillar? No. Matter of fact, if I hadn't watched the entire process from start to finish, I would have laughed you off the face of the planet. Because clearly there's no way in the world that the caterpillar, a worm, is transforming into something like the butterfly. So here is diagnostic evidence of one living thing transforming into another living thing. So is it such a logical leap for a non-living thing to transform into a living thing? Logically, no. If there's a mechanism for it. So you can see how rationally people can actually sign on to this and be like, listen, it's a rational hypothesis, but you have to debunk it. And so far you haven't, right? Now notice, the reason why I love spontaneous generation, because I always love to hit science right in the gut. It's always good to do that. Think about this. There is a good, rational reason to believe this, wasn't there? Think about it. This rock wasn't just this rock. It was reinforced, by the way. Think about it. You have meat sitting on the table, and then all of a sudden you see little worms starting to crawl around it. Where did they come from? Right? I mean, you didn't see any worms crawling up to it. You were watching it, right? They just seemed to come out of nowhere. Remember what happened. This is always based on your ignorance, or our ideas are based on our ignorance, not on our knowledge. Notice what happens in this particular case. We don't understand entomology. We don't know the life cycle of the housefly. We don't understand that the fly that's crawling around on the meat is laying eggs that we can't see that then become the worm. So we don't know any of the embryology of fruit flies or house flies or anything of that nature. So for us, that knowledge is not there. So when you consider that, your basis of ignorance, is it a rational hypothesis to think that, well, the meat just transformed into these worms somehow? Yeah, it's rational. Was it correct? No. The reason why I love this idea is because for all of its glory, spontaneous generation was not a Fruit Loop idea hatched up by crackpots. Spontaneous generation was a well thought out, rationalized hypothesis that had staying power over observations for quite a long time. There were good arguments and observations and experiments that supported spontaneous generation. So for all the world, to somebody in this era, it looked like spontaneous generation was right. As it turned out, it was wrong. I always like to tell the story because oftentimes this is what happens when you fall in love with your idea. Remember our first conversation at the beginning of the semester, never fall in love with your idea. And never get so dogmatic about your hypothesis that you start to presume that it is true when it is not. Right? Remember all those things where we say it's like the more you model, like when you go back to the endosymbiosis theory, it's so far back that, yeah, does it sound good? Does it explain a lot? Yeah, it does. Do we think it's the right way? Yeah, we do. But you know what? We used to think spontaneous generation was correct too, and it turned out to be wrong. At some point, we always do this to ourselves. We do a certain series of experiments and we realize, oh, wait a minute. So we were totally wrong about this. Yes, you were. Right? Like the latest one that I love is the whole T-Rex thing, right? I started off the semester with that one too. Right? We used to think that the mineralization process was a certain way, but obviously it's not the way we thought it was at all. If you have viable protein in a bone that's supposed to be 65 million years old, right? What that basically says is, listen, what you used to think was right isn't right. And you finally did an experiment that allowed you to be able to see that, right? And so that's the reason why I love this story, is because we had to persist at this. And this one looked really good for a long time. Okay, let's take a look at some of the other ones. So blending inheritance. Versus particulate inheritance. So basically what happened with the blenders is they basically believe that your traits blended together. So here's what they did. So what they did is they took snapdragons. As a matter of fact, I think Darwin was working on this. So even Darwin knew evolution was a genetic story. He just couldn't get it to work out. So we're going to take some snapdragons and we're going to cross them to each other. Now, this is not what a snapdragon looks like. If you have a snapdragon, they're irregular in shape. I can't draw them. This is my snapdragon. So this is all I can draw. So basically what's going to happen is you're going to take a red snapdragon that is a true breeder for red, cross it to a white snapdragon, which is a true breeder for white, just like we did in the genetics lab. And when you cross it, your offspring are pink. And then when you cross the pink to a pink, that's a pretty shameful looking flower there. Just go with it. What you got was red, white, and pink. Okay, first of all, let's take a look at this. So with the blenders, what they did is they saw this, right? They saw you add red and you add white and you get what? Pink? Well, wait a minute. Does this sound familiar? Sure. If you add red paint to white paint, what do you get? Pink. So what did you do? You blended it, right? So here's what the blenders thought. They thought, well, basically, wait a minute. What you're doing is you're actually blending your traits, right? Well, here's the problem where the blenders kind of fell apart. So they were able to explain this third phenotype, right? This third look. What they were not able to explain was how you got your original colors back. They couldn't explain that one. So that's kind of where Darwin sort of fell apart. Think about this. A lot of times people always say, it's like, well, the blenders didn't really believe like it was paint. And that's not what they were thinking. No, that's exactly what they were thinking. You know why? Because we can actually see evidence of this in anatomy and physiology. Because what we used to believe back in those days was that your inheritance was actually a blending of what we refer to as the humors, which is essentially roughly body fluids. As a matter of fact, the term humor is something that persists in the English language today. And it's a relic of this idea. So ultimately, we believed that the fluids of the male were mixed with the fluids of the female, and that created a blending of the female traits, the female fluids, the female traits, and the male traits with the male fluids. And you blend it together, just like paint, exactly like paint. And you got this sort of mixed outcome, pink. So they actually did believe that this was like paint mixing. But here's the problem. If I take blue paint and yellow paint and I mix it together, what do I get? Green. If I take the green and mix it with green, what do I get? Yeah, do I ever get my blue back? No, it's gone forever, isn't it? Well, see, this is where the blenders fell off. They couldn't explain this down here. Because what the blenders saw was that when I mixed green and green, I got green, but then I also got blue and yellow. So they couldn't understand how, once you mix it, can you get back your individuals? That shouldn't be possible. So that's kind of where the blenders were. By the way, this was something that permeates all of physiology in the 19th century, and actually before the 19th century. This is, by the way, a lot of what we do in 19th century medicine, like those ridiculous things like bloodletting, for instance. Because, yeah, and none of that, but mental health, that was a great one, right? And we still actually have relics of that, right? So here's a good example. I'm going to ask you two questions. Melvina probably already knows the answer to these. Right, so I'm going to ask you two questions, and I want you to tell me what I'm asking you. Do you have a good sense of humor? What am I asking you? What? Yeah, can you laugh at a joke, right? Can you take a joke, right? So, and then the second question is, are you in good humor? It's a slightly different question. What am I asking you? Are you healthy, Megan? Yeah, in the 19th century, that would have been certainly so. But when we ask somebody that, are you in a good humor? In today's world, we're asking what? Not can you tell jokes? Something a little more specific. Yes, exactly. Thank you, Megan. Yes. Are you in a good mood? Right? Why? Because in the 19th century, in this time, we used to think that all of our sort of psychiatric realities, our moods, our mental illness, were all a function of our body fluids. So if we ask somebody, do you have a good sense of humor? We're asking, do you have body fluids conducive to being able to laugh and take a joke? So all of your moods and your personality and your emotions were thought to be part of your body fluids, your humors. So if I'm asking, are you in good humor? I'm asking, are you in a good mood? Are you in a good mental state? Is your mental health good? Right? Because that was driven by the humors. And not only that, but the humors were the source of everything good and bad. So for instance, if you got sick, then the idea was that your humors were off and that there was some sort of a negative or bad component in your humors, and that all they had to do was just basically expel or eliminate that bad stuff from your humors, and then you would be able to kind of flush it out. So that's the reason why they did bloodletting. So they'd basically just tie you up, they'd cut an artery or vein, and then what they're doing when they're bleeding you out is they're trying to bleed out all the bad humors, thinking that they're getting out all the bad stuff. And then, of course, they patch you back up, and then they allow you to sort of rebuild yourself. So actually bloodletting wasn't some barbaric, crazy quackery stuff. It was based on this idea, which actually had some rationale behind it. But that's exactly how the blenders were thinking. They were thinking in terms of the humors, your body fluids, that these are how your traits are inherited, that they're mixed together as fluids. Now, a particulate individual believes slightly differently. They believe in discrete units or factors. So what they believed more was like saying, well, the red is red, not because of the fluids, but because there's like a set of instructions, like a packet of instructions that says redness. And white is white because it has a white packet of instructions. So when you mix the two together, well, this is where the particulate people couldn't explain it. They're not sure how you get pink, right? Because if I've got white instructions and I've got red instructions, pink to me seems like a completely alternate set of instructions, like a totally different set of instructions that came out of nowhere. So where did they come from, right? Because all I've got is white and pink. So I mean white and red. So how do I get a completely third, what looks to me like unrelated set of instructions, right? This is where they fell off. The blenders could explain this first step. The particulate people couldn't. The particulate people could explain the second step, but the blenders couldn't, right? So for the particulate people, they say, well, wait a minute. We're okay with red and white coming back because if you still have those instructions in there, you should be able to reclaim them, right? But that basically was the argument that was happening between these two camps. And largely you had spontaneous generation kind of hovering in the background. Sperm men had been debunked probably the previous century. There might have been a few hangers on still with that one. But mostly you had this kind of war going on between the blenders and the particulates. Enter in some of the early work on genetics. So basically, starting in the 18th century, we start to build our genetic story. Now, first of all, the reason why I like this, and not all textbooks do this, not only because I'm a history buff and I like the history aspect of it, but because the one thing you always have to remember is that typically in society, we only celebrate a person for a particular discovery. We do it with the Nobel Prize, right? We have a person that we recognize for this great discovery. And we do it in our textbooks and we do it as a society, right? We think of whose big thing was the theory of relativity. And you all say? Relativity. Crazy hair. Great. Yeah, Einstein, right? So, but the thing is, was he the only one working on it? No, but he's the one we give all the credit to, don't we? Same thing when he won his Nobel Prize, he didn't win it for relativity. A lot of people don't realize that. He won it for the photoelectric effect. And we look back and we say, oh, Einstein's this great genius. He did relativity and he did photoelectric effect. And so he came up with it, but no, not really, because you had Max Planck, who was involved in photoelectric effect as well. A little known fact, Einstein had a whole bunch of colleagues working with him on relativity. He had his wife who was working with him on relativity as well. That's a very, very unsung hero there. So generally speaking, what we see in science is that there's never a person who just sort of somehow magically, sort of divinely gets some shock of inspiration. And, you know, it's like a night or day sort of a thing. Science is built progressively as a community. So when somebody makes a great discovery, that could only have happened because of all the infinite discoveries that went before them. It's like climbing stairs, right? So it's like you hail the person who's at the top of the stairs, but in reality, if they hadn't had other people behind them covering the first bottom step and the second step and the third step and the fourth step, they would have never gotten to the top step. Okay, so science is actually an amalgam of most of whom we never talk about. And one person, unfairly so, gets all the credit. That's just kind of the way we are. But this is the reason why I like this, because it shows us a little bit of that history. The Mendel didn't just come out of nowhere. Mendel stood on the shoulders of giants, just like we all do in science. Okay, no rhyming intended there. But anyway, let's start off with the first one, right? Kohlreuter is starting off in the 18th century. And so what Kohlreuter is doing, so Kohlreuter is basically a tobacco farmer, right? And back in the day, what you're doing is you're creating a crop. And so just like you're always doing with anybody, you're trying to create the best crop and you're trying to gobble up market share. There's economics associated with this, right? The same thing happens today in agriculture, right? You're trying to always produce the bigger, better, badder, more delicious products so that everybody buys your stuff and makes you lots of money, right? That's exactly what Kohlreuter was doing. He was basically in this for the money. And so what he was doing is he was doing what a lot of agricultural people do, is he was making strains of tobacco, trying to essentially find the ever most popular tobacco strain, right? And if you're kind of in a tobacconist shop, right, you'll notice there's a lot of that in there, right? Especially not necessarily, well, first of all, if you go into the cigarette aisle, you'll notice there's lots of different options there. Everybody's trying to say, I'm the best, right? But if you go into like a tobacco shop where you're actually buying the actual tobacco itself, I mean, you've got a myriad of choices there. All these folks are trying to say, my tobacco tastes better, it's better, you know, it's all of it. I mean, they're trying to prove to you that I have the better product, right? That's exactly what Kohlreuter is trying to do. So as he's producing these tobacco strains, what does he notice? Well, first of all, he noticed that when he produces the hybrids that he's producing, is that they're different from the parents. Now, this is important because when you're different from the parents, it basically contradicts and debunks direct transmission. So what does that mean? One of the early genetic ideas rattling around in this area was that in direct transmission was that what you got was essentially a carbon copy of what your parents had. Right, and that would be like direct transmission. So you would essentially get the exact same thing that your dad had, the exact same thing that your mom had. And so that would be basically a direct transmission from the parents to the offspring. And so the fact that the offspring were different than the parents proved that what I'm getting is not the same as my parents. So I'm not getting a direct carbon copy from my parents. That's what direct transmission is. Kohlreuter was the first one to actually technically essentially eliminate that as an option off the table. However, also what Kohlreuter noticed is that when he crossed those hybrids together to create the second generation, that is to say the grandkids, essentially, he noticed increasing variation there. So this is important if you're trying to play this sort of tobacco strain game, because some of those variations may be marketably beneficial for you, right? You may be interested in actually pulling some of those together and creating more of that particular type of tobacco that you see in the second generation. So that's what kind of Kohlreuter basically added to it. Now, T.A. Knight really basically had a lot to do the direct lead up to Mendel, because a lot of people don't realize this, but T.A. Knight in the early 19th century, notice when he was doing his work, he was doing his work in the early 19th century. So we're talking Beethoven's still alive at this point, not for very long. I think he died in 1824. But you're only about maybe a little more than 10 years away from Schleiden and Schwann's cell theory. You're about maybe 40 or so years, a little over 40 years away from theory of evolution, Darwin's paper, right? The transmutation of species by natural selection. So you're about 40 or so years away from that. At about 1823, I believe this is the time frame right in around here. Not exactly sure what the exact date is that Darwin is actually going on his little around the world trip on the Beagle, where he's collecting all of his stuff. So that's basically what's happening at this particular point. T.A. Knight is basically using garden peas, Pisum sativa. And he does a couple of things, really, really crucial things. And this really set up Mendel. And this is a perfect example of some really critical work that Knight did that made it possible for Mendel to do his work. First of all, he's the one who created and figured out how to generate true breeding strains, right? So remember we talked about true breeding strains. We talked about that with the corn plants. Remember we talked about the genetics. And we said that a true breeding strain is basically a strain that's able to breed true for its traits. So for instance, if you have yellow corn, then that means if I take that yellow corn and cross it to a yellow corn, it will give me yellow corn, right? If I take that yellow and cross it to a yellow, it gives me yellow. That means that corn is true breeder for yellow. So whatever is in there, like if you're talking about packets of instructions, it only has the instructions for yellowness, and that's all it has to give. Okay. Now it's important because what doing this does is it minimizes variables. So you don't have to worry about multiple instructions in there. You have one set of instructions. So it really streamlines the situation. So what T.A. Knight did was basically took two true breeding strains, similar to what we saw in the corn plants, and crossed them together. And then asked, what do I get in the next generation? That is to say, my first familial generation. So in this particular one, we call this the parental generation, or P. Then you cross the parental true breeders together to create your F1. This is your first filial generation, or F1. And then what he noticed was that you only resembled one of the parent strains. Well, that's interesting, right? Because you had two sets of genetic information going in. So you only look like one. So there's a head scratcher, right? What happened to the rest of it? We're not quite sure what happened there. So this was a little bit of a barrier for T.A. Knight, wasn't able to figure this one out. But then what he did was basically he crossed the F1s to each other to create the second, or the F2. That's the second filial generation. And then what he noticed was he generated all three. He generated the parentals and also the F1. So he had both. So if you think about it, actually T.A. Knight was actually onto something. He was just never really able to figure it out. So this was the work leading up to Mendel. So a lot of times when you take a look at Mendel, basically he chose pea plants. Why? Probably because he was familiar with T.A. Knight's work. What else is a really good choice about pea plants? Well, this is true for all genetic organisms. First of all, you got lots of varieties, right? So you got a lot of material to play with, so that's always helpful. They're easy to grow, right? You have to, I mean, if they're hard to grow, then this is not a good genetic model organism. Not only that, but you can actually self-fertilize them versus cross-fertilize them. So corn can do that as well, by the way, right? So if you take a corn plant, there's both female and male parts to it, right? The tassels represent the male pollen, that's the male part. The actual corn stalk, which is actually, it's not corn once it gets there, but it's kind of like rudimentary. That's the female part. So one corn plant can actually fertilize itself. That's called self-fertilization. Plants can do that. We can't, but plants can do that. They can also cross-fertilize, right? So for instance, if my tassel blows in the wind, I can fertilize the plant next to me as well. So I can both do self-fertilization and cross-fertilization. And that's true for pea plants, and that's critical. So here's the dude himself, right? Our favorite Austrian monk. And so a lot of times what'll happen is we kind of tend to sort of deify Mendel a little bit sometimes, and Einstein as well, right? We kind of think, oh, well, there's this kind of like, monastic, I don't know, I'm not Catholic, so I don't really know a whole lot about, monks and stuff like that. But I do know that relatively the monastic tradition is one of seclusion oftentimes. And then generally speaking, monks don't do a whole lot. I mean, they I guess they pray and study and things like that, but they're oftentimes, not always, but oftentimes disconnected from society. And so here, you kind of have this image of this sort of monastic sort of individual, almost hermetic in nature, rattling around up in the Austrian Alps in a monastery. And then he gets this divine spark of inspiration from on high. And then all of a sudden he figures out these genetic principles, and it's like, miraculous, right? We think, by the way, all of our major achievers, we do that too. Einstein, we do that too, right? How many of you have heard, oh, well, Einstein failed algebra? Right, okay, first of all, Einstein was never not a genius, right? Or how about this one? Oh, Einstein was a patent clerk. Okay, so it wasn't like Einstein was working at the post office and then all of a sudden he kind of got this sort of lottery-like divine inspiration and became a physics genius. Einstein was always a genius from beginning to end. It didn't just happen randomly. Mozart was always a genius from beginning to end. It didn't just happen miraculously. It was just something that he was. Mendel was a product of basically the same system that we have. Right? A lot of times students will be like, well, what did Mendel look like? And Einstein, for instance, if you want to demythologize Einstein, just study all the other great German physicists that he worked with. I mean, there's a lot of them there, right? I mean, you have Schrodinger in there, Planck was alive in those, Niels Bohr. I mean, you have a lot of really heavyweight, some of the greatest minds ever actually in human history were all peers and colleagues of Einstein. When you study the stuff that they were doing, all of a sudden Einstein kind of looks like he's just one member of the team. I mean, there was a lot of great brain power in that era. They all came from Germany in that area for some reason. It's just, I guess the Germans just hit the jackpot on physics during that generation, apparently. But when you take a look at it, Mendel can be best thought of, not like this kind of monastic hermit, but actually Mendel, a lot of times students say, well, what was Mendel like? Well, he was probably most likely what we would think of as a university-trained research professor at Boulder. So think of somebody, a professor at Boulder with a research lab. Those folks are very well trained, very well educated, very well read. All those professors know exactly what's going on in their industry, in their field. So they know who's doing what and what results they got. They oftentimes are also well connected. They know the people who are doing the research. And they oftentimes know them personally, right? So they're also well connected. So when somebody makes a great discovery of Boulder, we're all like, oh, well, that's not really all that surprising because they're so well educated and so well trained and so well connected that that's kind of what they do. Mendel was the same way, right? A lot of people think that Mendel was sort of the secluded hermit sort of a guy. It's like, no, that's not true. Mendel knew about Darwin. He knew what the things were Darwin's working on. He knew about Colruyter's work. He knew about T.A. Knight's work. He knew about all these people and they knew him. So he was already well read and well aware of what was going on in the world. So he was like a well-trained university professor, a research professor. So the reason he chose pea plants was because he saw what a good system it was that T.A. Knight had when T.A. Knight was doing his work. So let's take a look at what Mendel actually did then. So basically what Mendel did when we did his experiments is he basically did what T.A. Knight did. So he basically produced these true breeding strains for different types of traits. So he did lots of different traits. And then what he did was basically he produced what's called reciprocal crosses. So for instance, if you take a look at a reciprocal cross, this is something that T.A. Knight didn't do. But let's say, for instance, that you're going to take a flower that's red and cross it to one that's white. If you have a male red to a female white, that's one direction. But then the reciprocal would be a female red to a male white. So what does this do? Is gender a factor? Right, is the parent of origin a factor? Doesn't matter where red comes from. Is it different if it comes from dad than when it comes from mom? Nobody's asked those questions before, right? This is one of the things that really sets Mendel apart. Nobody asked this kind of question before, right? So they never even thought about that. Then what he did was basically he created, just like T.A. Knight, he created his hybrids, and then he crossed those together by creating his F2 plants, or second filial generation. So let's take a look and see what one of the benefits of this cross-fertilization versus self-fertilization gives us. This is kind of what Mendel did, right? This is one of the things you want to have in a genetic organism, is you want to be able to dictate how it's actually crossing. For instance, you want individual A to mate with individual B because you want to know what the outcome is there. Well, here's the problem. Some organisms are going to be amenable to that. Us, for instance, we're not very amenable to that, are we? we tend not to like that people to say, listen, I want you to mate with that person over there. It's like, I don't really know that person over there. And I'm not even attracted to that person over there. So no, right? We like to have some say in it, which makes us a terrible model organism, right? So that's one of the things you look for in a model, in a genetic organism, is you have to be able to manipulate the mating. And this is the way you do it with pea plants. And actually, by the way, this is a technique that you still use in horticulture all the time. So whenever you have horticulturists producing hybrids, this is exactly what they're doing. So let's take a look at the structure of a flower here. So here's your flower. You have two different parts to it. So the anthers basically are the male areas. So this is basically the areas that have the pollen, which is kind of like the male gamete. And then what's going to happen is you're going to have what's called the carpel. Now, the carpel basically is going to compose of two different pieces. I'm going to write mine sort of as a picture. So the top part here is what's called the stigma. It tends to be sticky. It's kind of the top of the picture. And then basically what you're going to have is the style, which is the neck of the picture. And then underneath here are going to be all the little eggs that the female portion of the plant has produced. Now, generally what happens is a pollen will blow or be delivered by a pollinator to the sticky stigma. And then when it does that, what it does, it goes through mitosis and creates what's called a pollen tube. Which is like a little channel or tunnel for that little male gamete, the male nucleus, to sort of move down. And then what'll happen is it'll fuse with this little chamber, which has the egg in it, and then it'll fuse with the egg, and that'll basically create the zygote. And so you'll have, multiple pollen grains on there. So these multiple tubes going down in there, connecting to each one of these. So here's the problem. Pollen and pollination is random, right? So if it's windborne, then basically you're going to have pollen just randomly blowing all over the wind, right? So that's not a good thing. So what happens if I want to take a purple individual here and mate it specifically to a white individual? So that is to say a purple female to a white male. How do I do that? Right? By the way, pollinators are a problem too, aren't they? They just walk around, collect pollen, and they just walk all over the place. So how can you get intentional pollination? Well, what you do is you do this trick. So for the female recipient, all you want are the ova, the carpel. So what you do is you basically cut off the anthers. So there's only the female part left. That ensures that all you have are purple eggs, no longer purple pollen, because you cut them off. Then what you do is you take a paintbrush and you brush off the pollen off of your white male donor, the pollen grains, and then you brush it on the tip of the stigma. That way you can get white pollen to purple stigma. And that will give you your cross, your intentional cross. Purple female to white male. And basically this is how you do controlled breeding in horticulture, actually, throughout all of horticulture. It happened with Mendel, and we still do it today. Okay. So let's go ahead and take a look at some of the things that Mendel did. So first of all, one of the first things he did was what's called the monohybrid cross. And we took a look at that in lab a little bit. What we want to do is take a closer look at it. And so what he did, first of all, was he established true breeding strains, right? He did 7 different traits. And the important thing about these traits is there's only 2 variants, right? This is what we call a binary trait. Here's a good example. Either you are white flower or purple flower. There's no in between. You're either tall or you're short. There's no in between. You're either yellow or you're green. There's no in between. So this is what we mean by binary traits. You're either A or you're B. There is no in between. Now, what does this do? This is important. Because it eliminates the intermediate phenotype. That pink, right? Remember what we got with Snapdragons? Red and white gets pink. So it eliminates that third piece. So it's either red or it's white. There's no in between. Okay. So that was the important thing. So then what he did then was he crossed his parental generation together to get his first filial generation. And he noticed the same thing that Kallreuter noticed, was that it only resembled one parent. And then ultimately, there was only one, in this case, like for instance, if you're using plant color, right? The look that he saw was purple. The one that he didn't saw that disappeared was white. And then the other thing that he did was he crossed those two F1s together to create the F2 generation. And then what he saw was a ratio of 3-to-1, 3 purples to 1 white. And this was a common trait. So he saw this. For all 7 traits. 3 purples to 1 white, 3 greens to 1 yellow, 3 talls to 1 short. He saw this over and over and over again. So this is what he saw. So you notice purple to white, 3.15 to 1. You got yellow green, 3.01 to 1. So still 3 to 1, right? Brown wrinkled, 2.96 to 1, 2.82 to 1 for pod color, for pod shape, 2.95 to 1, pod flower position, terminal versus axial, 3.14 to 1. And of course, tall versus short was 2.84 to 1. Notice in all cases, you're not exactly 3, are you? But you're pretty much there. You're pretty close to 3 to 1. So notice though, 3 to 1 is our theoretical, yes? But the actual numbers he was seeing were not spot on 3 to 1, but they were close to that, right? So that's basically what Mendel saw. So one of the things I want to do, however, and I'm just going to get this started and set up. Is we're going to talk about the monohybrid cross. And what I want to do is I want to compare this and foil this against scientific method. I'm going to do these side by side. And what I want to do is I want to start off with this one. And I want to do this exactly as Mendel saw it, not with all the answers in the book that we have now. But I want us to take the approach of the way Mendel saw it, because there's a couple of things that I want to tease out. And there's a few slides that I'm going to be adding in here. Okay. However, we're at our witching hour. And so that will be a tale for not Wednesday, right? Because Wednesday is exam day. And that'll cover 5, 10, and 11. Also remember that Wednesday is also our case study day, which means it's a day off for us because we're on target. So it's a free lab day. So I mean, not just you can go home after the exam is what I'm saying, if you don't have other obligations afterwards. So that's basically how Wednesday is going to take a look. Does that sound good?